Okay, so thank you guys so much for being here today. I'm really excited about this next panel. We're going to have talks from 3 leading biopharma companies that are currently conducting phase 3 clinical trials for which GRIN disorder patients are eligible. And so today we have these representatives here from GRIN Therapeutics, Longboard and Lundbeck Pharmaceuticals, and Praxis. And they're going to be sharing with you guys some critical updates about their pivotal studies. This is a really unique opportunity for you guys to all learn about the progress, the potential, and the next steps for these late-stage trials, which have significant impact on our community and makes important strides towards new treatments for GRIN and for the GRIN community. And so I want to start by welcoming Dr. Mike Panzara from GRIN Therapeutics. Thank you for being here.
All right. Well, thank you very much for the intro, and thanks to GRIN for inviting us to participate in this session. I said I'm Mike Panzara. I'm Chief Medical Officer of GRIN Therapeutics, Nervati Neurosciences. Neurologist, been working in the business for 20 years, but never in this area. And this is my third GRICon, so it's exciting to be here and to share where we are, because the initial one that we participated in, we were just talking about the idea of this, and we've made some great progress, and thanks mostly to the support and the participation of the community, which I'll go through. So we're just going to talk just very briefly about the company at this point, because as I said, we've been here a few times now, and then go through where we are with the program. So we're based in New York. And actually, the company has its roots to the 2019 conference at Emory University, GRIN conference at Emory University, and then basically through funding from Blackstone Life Sciences, the company was born. And essentially, it's really been a partnership ever since then. This just has a timeline of where we've been throughout our life cycle and the number of interactions and touch points in the formation of the company and the formation of the program. It has been really an exciting and collaborative time. I mean, we were here at the time when the company formed. We were here at the time the study was designed. We've had a partnership in our discussions about phase 3, sharing of the phase 2 data, our interactions with the regulatory authorities. We've gotten feedback from the various members of the community around the world along the way. So it really has been an exciting partnership, and which makes it even more makes us even more really thrilled to be here to sort of say where we are. So a little bit about why we're evaluating radiprotil in GRIN NDD. For those, you could see a big picture of the chickens out at the table outside, but this is a little bit of an example. And we've been using this example for a few years now, thinking about the NMDA receptor as like a gate on a fence. And you've heard other analogies of the receptor during this meeting. This is our sort of way of thinking about it and illustrating it, is that you have this gate on the membrane that allows flow in and out. And when you have like almost, the gates holding the chickens in. Gain of function, too much flow, gate breaks, chickens run out. Loss of function, poor flow. It just leads to a dysregulation or an abnormal flow through the membrane, which leads to the signs and symptoms we're all very familiar with. I don't have to tell this group that there are no approved treatments for GRIN, but we are hopeful based on the mechanism of radiprotil. There is potential for there to be modulation and modification of the disease. You have, again, illustrations on the right here of the NMDA receptor. And the pathogenic, this illustrates the pathogenic receptor, where you have the binding of radiprotil to the GLU-N2B or the NR2B subunit. And it's this idea that when you are targeting the abnormal subunit of the receptor, you can change the flow and the conduction of the receptor which then would influence the signs and symptoms of the disease, like seizures and some of the behaviors, and then over time that would lead to an effect on neural development. So that's the story and the approach behind the use of Radiprodil, which, as you heard, there are different tools. This is a small molecule, so this is an oral medication that's taken twice a day, and it's this small molecule that's binding, this chemical compound, small molecule that's binding to the receptor. So we have a variety of studies ongoing in GRIN and DD. The key study that has led us to where we are today is the phase 1b study in gain-of-function patients. So the idea, GRIN is, Radiprodil is what's called a negative modulator, a negative allosteric modulator. So the idea would be it would potentially be a little bit more effective in gain-of-function, kids with gain-of-function variants. So that's where we started. We started off with a gain-of-function study, which was open label, and this was over a period of 8 weeks, patients with and without countable seizures, because we wanted to make sure that we could assess the impact on the types of seizures that are easily counted by caregivers to give us a sense of activity. But we didn't want to exclude patients without seizures or without countable seizures beyond a threshold. So we wanted to assess both. And actually, the study looked good. The drug was well tolerated over the course of the study, and we had most common types of adverse events were those that are associated with infections. We saw infections, and we also saw symptoms described of the disease. But it's open label, so it's difficult to determine what's related to drug and not. In the motor seizure cohort, there were 2 cohorts in that study. We saw 86% reduction in these countable motor seizures in patients who came into the study having tried and failed on at least 2 seizure medications and had to have at least 4 countable seizures to come into the study. So in that group, there was about 86% reduction in the seizure frequency, and most patients had at least a 50% reduction. So there's quite a remarkable result in these 7 patients that were in the study in the countable seizure, with 6 of 7 of them having 80% of the days seizure-free during the 8-week observation period in the study. So this was a striking result even for us. This is beyond what we anticipated and beyond what we were planning to do, because typically at this point you'd go into sort of a phase 2 study, because this was a phase 1b primarily. In addition, we saw some promising effects on what clinician and caregiver assessments of well-being. So these global clinical assessments, asking physicians and caregivers, have you seen an improvement? And we saw this improvement, as you might expect when you're reducing seizures, but we also saw improvement in those without seizures. There was something that's happening in this population that suggested a benefit of some sort, a positive change, regardless of the occurrence of seizures. Acknowledging this was a small number of patients, and it was an open label study. But it was enough to provide some encouragement that there may be an effect. And then when we saw these effects, as I said, they exceeded our expectations. So we engaged the regulatory authorities in the US and Europe. And the regulatory authorities also thought this was a little bit more than they anticipated. And that's why we shared this. They authorized us to go from a phase 1 study directly to a phase 3 study. And that's pretty unusual. Normally there's phase 1, phase 2, phase 3. I've presented at this meeting before about the phases of drug development. But sometimes, as I've said in those presentations, you can combine phases and jump phases. Have never done a jump from phase 1 to phase 3 before, but these data in support of the authorities was felt to warrant that. In addition, we've gotten breakthrough designation, which in an open label study is pretty unusual, and also orphan designation and another designation in Europe called PRIME, which is sort of another accelerated designation. So it's been a really exciting time to see these data in these small data set the effect that has been seen has encouraged, has at least engaged the authorities to help us figure out how we move this forward in an appropriate way. And throughout the time, we've also had our independent data safety monitoring committee reviewing all the data to determine whether there's any change in the benefit risk, and there hasn't. And the most recent assessment of that was in June. So phase 3 B-line study, that is our phase 3 study that we're getting ready to initiate. We will enroll, just like we have before, patients with and without countable motor seizures. What has helped us accelerate this program is the amending of the phase 3 to phase 1 to a phase 3. So this has allowed us to take the same concept of our design, but add some important elements. Broadening the age of the same type of patients with GRIN gain of function variants, countable motor seizures, who have tried other therapies. The non-qualifying seizure cohort, again, no seizure threshold requirement, but a severity requirement on the non-seizure symptoms. Importantly, many studies, in order to get in, and those who've tried to get into other studies you need to reach a certain seizure threshold, otherwise you can't get into the study. Here, for one cohort that's true, the other cohort's not. So it's really open. If you're within that age range and you have GRIN and you meet behavioral and seizure criteria, there's a place in the study. So we're hoping that also allows us to assess both basically the entire disease spectrum of GRIN and DD, and also open the opportunity for more participation. There's going to be concomitant drugs. There are certain drugs I think you've seen in other presentations here. There are other drugs that target the receptor, and you've heard about different receptors. Obviously, can't be on those drugs during the study, otherwise that's going to make it difficult to understand what Radiprodil is doing. As with any study, there's usually a whole host of other exclusion and inclusion criteria related to the other medical problems, et cetera. Importantly, one of the biggest things we've done in modifying the study was to add a placebo group. And this is actually just something that in the regulatory setting cannot be avoided. It's very important, especially with some of the behaviors and symptoms. Sometimes it's hard to know what's related to the drug, what's not related to the drug, what's related to disease. What we've done is, though, we've negotiated as minimum time possible for the placebo group, which would be 12 weeks of placebo in the seizure cohort, and you can see that here, 12 weeks for the placebo and active placebo group in the seizure cohort, and then 24 weeks of placebo in the non-seizure cohort. But importantly, at the end of the study, all participants in placebo and active groups will have access to long-term Radiprodil, assuming the benefit risk supports it. Obviously, if it's not going to work, we wouldn't want people to access. But if we're seeing benefit, if we're continuing, the idea would be to continue access to the drug until it's actually approved and reimbursed in the countries in which we're working. And you can see one of the challenges here is going to be these number of patients. So we're looking for up to 60 in the seizure cohort and up to 40 in the non-seizure cohort. So a total of 100 people for the study. Now, endpoints. We had mentioned the seizure cohort. So obviously in the seizure cohort, your primary endpoint is going to be change in seizures. So this is countable motor seizures. Certain criteria defined by the Epilepsy Consortium. There'll be a limited number of the types of seizures. This is very typical of these epilepsy studies. The non-qualifying seizure cohort, safety is the primary endpoint, but we're also, importantly, the key secondary endpoints here in both cohorts are exclusively focusing on behavior. So are we changing the behaviors with treatment? And the behaviors we're looking at, you're seeing the ABCC irritability subscores, what we're using, as well as some of the other typical symptoms that you all know so well. These are all driven from a survey that we did with the community by a company called Lumanity, where we were able to collect the most important seizures to the community, most important symptoms, signs and symptoms to the community, and then figure out what tools we can put into the study that actually measure those signs and symptoms so that we can then measure an effect on those signs and symptoms. And that's what you're seeing here, where we not only are collecting those symptoms to focus on the ABCC irritability scale, which captures a lot of those, but we also are working to develop a GRIN-specific clinical outcome assessment. So the way this is done is, again, we started with that survey. We collected those most important signs and symptoms. We're getting it down to a few key signs and symptoms. You can see there these core domains that were felt to be most important: receptive communication, expressive communication, gross motor skill, fine motor skill, and just dysregulated behaviors. And those aspects will be put into this model, what's called the conceptual model, where we have, again, will create a scale that then the clinicians could apply against these important signs and symptoms. And we'll be deploying that and validating that for the first time in the study. But once it's done, the CGI, the GRIN-specific CGI that comes out of a study like this, then hopefully, if it is validated and is workable, is something that can be applied in other studies. So again, we've taken the community's views, we're incorporating it here, and hopefully the goal would be to have a tool that we can give back. The other thing that's happening in parallel to this is we're starting up a natural history study. So we're actually, even if a patient doesn't qualify for the inclusion criteria, for our study, we're going to have a study that they can go into where there's no intervention at the current time, but at least we can monitor and understand more about the disease. This caregiver assessment, this the ABCC, seizures, et cetera. This is a place for loss of function, place for people who are indeterminate function, as well as gain of function who might not qualify for the study. Again, the hope is to then have that in parallel with the treatment trial and the placebo group in parallel, and create a story about what is that natural history of GRIN that we can then use the other registries that you've been hearing about during this meeting, and then apply that to learnings for the disease that can advance other therapeutics for the disease, some of the exciting future therapeutics that we've been hearing about. So in summary, mechanistically, we believe that that with a drug that modulates the NMDA receptor and the core biology of the disease, we think that the approach has the potential to have really a meaningful impact in the disease by affecting the pathway. We're going to begin enrolling patients mid to end. We're sort of mid now, so we're second half of the year. We're just on the cusp. We're trying to develop tools that can broaden our understanding of the disease and broaden our understanding of the effect of the disease. And we have our natural history study that I just mentioned will be starting off in parallel. So basically, there's ways to get involved here. Understanding about your variant is going to be important to understanding if there's an eligibility, if you're eligible. We actually have a company called Informed DNA that we've partnered with that will help you through that process of variant identification and genetic counseling. It's a team of genetic counselors that will help you understand a bit more, and if appropriate, refer you on from there. Stay connected with groups like GRIN, GRIN2B, others who have really driven the progress in this area. And then there's our link right there. And I can't say any more than thank you. I can't thank you guys any more for what you've contributed. Opportunity to speak today, of course, but what scientists and investigators and collaborators and caregivers and everyone who has gotten us to this point, and we're looking forward to really hopefully bringing this to the next step and hopefully bringing a treatment to the community. Thank you very much.
Thank you so much, Mike. I'm sure you guys all have tons of questions, and we will have time for a Q&A at the end. But now I want to go ahead and welcome John Messina from Lundbeck Pharmaceuticals. Welcome. Thank you for being here.
Thank you very much. Thank you for having me. My name is, as she said, my name is John Messina. I head up medical affairs at Lundbeck Pharmaceuticals. I've been in the drug development business for about 30 years now. My background is I'm a clinical pharmacist with some postgraduate research on how to design and run clinical trials primarily. This is the first time I've been to a patient advocacy group meeting. It's one of the more inspiring things I've ever done, I'll be honest with you. This is fantastic. And I'm so excited and proud to be here. So thank you for having me. Bexicaserin I'm here to talk about, which is a molecule that was actually begun the development with a company called Longboard Pharmaceuticals. Longboard Pharmaceuticals and Lundbeck Pharmaceuticals has merged in the last year. And we've brought in that team from Longboard and are now part of our team, and we couldn't be more excited. They have really done a tremendous amount of work here in this space, and we're very happy to be part of it. A little bit about Lundbeck, though, as I get started, because I think it's important for maybe you to understand, you may or may not know that Lundbeck has been involved in the epilepsy space in the past. We developed or we marketed a drug called Onfi, which is clobazam, which was the first drug actually approved for Lennox-Gastaut in the United States. And since that drug went off patent, we had been, as a company, been looking for another opportunity to move into this, back into this space. The bexicaserin opportunity came along, and it's something that, because we were so impressed with the science behind it, we wanted it to become part of our company. So Lundbeck, if you don't know, we are based in Copenhagen, Denmark. We are a neuroscience-focused company. We are represented all throughout the world with offices throughout the world, and about 7 million patients per day will take one of the medications that we make at Lundbeck. When it comes to bexicaserin, I want to kind of jump in here a little bit and kind of talk about the mechanism that we believe is how it's going to be working and why we think it's a special molecule and why we think it's important. Bexicaserin is an investigational oral superagonist of 5-HT2C receptor, the serotonin receptor. I apologize for all of the abbreviations. We in pharma love abbreviations. And I'm going to try and, the next version of these slides, I'm going to try and take out a lot of these abbreviations if we can, just to make it a little bit easier to understand what we're talking about. But it's the serotonin receptor. Serotonin is is a neurotransmitter but it's also ubiquitous throughout the body in many places. The key thing about the 5-HT2C receptor is that it is really dominant within the CNS on these interneurons, which are GABAergic. GABAergic means they produce GABA. GABA is a brake on the system. It is a suppressant. And why is that important? Because when we're dealing with epilepsy and we're dealing with seizures, we're trying to quiet things down, right? We're trying to reduce that excitability. And by us actually, if you can stimulate those interneurons to release more GABA, we can potentially reduce the seizure activity. We can reduce that excitability. And so people have been looking for a long time to actually find a serotonin agonist that is specific for that receptor. Serotonin itself, which is a natural compound in the brain, will hit all of the serotonin receptors. But we wanted something that was just going to hit that 5-HT2C. Why? Because if you have something that is nonspecific and you hit some of those other receptors, it carries with it side effects and other effects that you really don't want, we call off-target. So for instance, it can produce cardiac effects, GI effects, etc. Well, the medicinal chemists who were involved with the development of this had been working for over 20 years to try and get a more pure, if you will, stimulation of that 5-HT2C. And we call it a superagonist because what they were able to develop is the stimulation that it produces at the 5-HT2C is actually higher than you get with natural serotonin. And it really has very low, if any, off-target stimulation. So the purpose of this product or this medicine or this drug is to hit those interneurons to produce more GABA and therefore quiet down the seizure activity. So that's the promise, that's the idea. There is a phase 1b/2a study that was conducted by Longboard. It was called the Pacific Study. Participants were included who had DEEs, and you're all familiar with DEEs here. This was a study where they wanted to look broadly across not just patients with Lennox-Gastaut or Dravet, but they were including patients with any number of these conditions. Now, they oversampled or over-randomized, if you will, to active therapy versus placebo so that they could learn as much as they could on the patients who get put on active bexicaserin. This was a double-blind placebo-controlled trial. It had a titration period and then a maintenance period of 75 days. Now, the titration period allowed patients to go up to either 6, 9, or 12 milligrams up to where they could best tolerate. At the end of that double-blind period, there was a down titration, and all patients were put into what we call an open-label extension, where patients get active therapy. And those patients were in a follow-up period for a long period of time. In fact, they're still ongoing today. The patients who we included in this trial had DEEs with an average of 4 motor seizures per the 4-week period during the 12-week prior to being brought into the study, and 4 motor seizures in the 4-week period at screening. So we needed enough seizures there to be able to show that we can improve that with medicine. But we did, as I'd indicated, patients were allowed with multiple syndromes, including LGS, Dravet, SCN2A-related epilepsy, CDD, among a number of others. We excluded patients who were on fenfluramine and loccaserin. We also, this was a US-only study with 34 sites, and it was adolescents to adults at this phase. The primary objective here was to see if we could reduce countable seizures in this patient population and identify potential indications in pivotal studies. And there was also a pharmacokinetic, which is measuring the blood levels of the drug after it's being administered to this population. One key thing I want to point out is that there was no echocardiograms required in this study. And the reason for that is for that specificity that we have in terms of where the molecule is targeting. It's targeting the serotonin receptors in the brain. So first things first, safety. What did we see in this trial? So first thing we see is it's fairly well tolerated. The number one adverse event that we see is somnolence. Now, one of the interesting things that we learned about the somnolence is most of this occurred during the titration period. And we learned in this trial that the titration was probably going too fast. So we altered it when we actually put patients into the open label period and slowed it down slightly, and it actually improved it greatly. So most of the dropouts that we saw in titration were because of the somnolence. When we looked at the placebo patients that went into that open label and using the slower titration, none of them dropped out. So we feel that by slowing it down, we actually improved the tolerability quite a bit. There was a few SAEs, and one I wanted to point out here is there was an ankle fracture in one individual. That was actually an individual who was wheelchair bound and was actually able to walk for the first time after they'd been in the trial, and because of their walking, they actually fractured their ankles. So obviously that fractured ankles is not good, but obviously we feel it's a positive thing in terms of if we can actually improve the patient's ability to be mobile, et cetera, something to hope for. Again, it's N of 1, phase 2 trial, it's still investigational, but again, it helps us as we move forward to the phase 3, getting an idea what we can anticipate. And regarding efficacy, so looking at seizure reductions. So if you look at the countable motor seizures, which was the primary outcome variable, we actually see about a 60% reduction in countable motor seizures after 12 weeks. Placebo group had a 17% reduction. If you look after that period, I think this is right, from here, this is the open label extension. So these placebo patients get put on active therapy. You can see that they, after another 3 months, get down to where the active patients were. If you just look at total seizures, so these are motor seizures, and this is including everything, including absence seizures, for instance, you actually see a similar reduction. So based on this data, the Longboard folks met with the Food and Drug Administration, have met with the EMA as well. The Food and Drug Administration had agreed with that based on this data, and because there was a large group of patients with multiple DEs included, that we could pursue an indication for DEE generally. It's something that they will have to evaluate at the end, but we can go for what we call a broad indication. To achieve that, our phase 3 program has to include 2 clinical trials, and we'll also have an open label extension. I'm going to go through some of the inclusion criteria for this trial, for these trials. But essentially, the first trial, Deep Ocean, this is going to enroll patients with DEEs generally. And the second trial is for Dravet syndrome. We want a broad group of patients so that we can get this broad indication. That's what's going to be required. All of these patients, when they successfully complete the double-blind placebo-controlled trial, will go into an open-label extension where they'll receive active therapy, and we can follow them for a long period of time because we want to look at long-term safety as well as efficacy, hopefully being maintained. In terms of the parameters, the primary endpoint here is reduction in countable motor seizures again. We will be including patients aged 2 years of age and older. There will be a weight-based dosing for those individuals. And then we will have there's 100 sites that will be available across the United States and the world for this. And we'll touch a little bit on it here, but we'll have more details for you out at the table if you're interested as we move forward. And we did get a breakthrough designation granted by the FDA for this. So let's talk a little bit about the study designs for these phase 3 trials, which are ongoing right now. And I'm going to focus on the left-hand side here, the Deep Ocean. So this is the DEE other. It includes patients with DEE and LGS as well. So we are going to randomize the LGS slightly differently, but they're included in this trial. And you can see the age of onset for inclusion is before 5 years of age for DEE other and before 8 years of age for LGS. In terms of the seizure types needed for inclusion, more than one of the following, GTC, tonic, colonic, atonic, truncal leg involvement, focal motor, including hemiclonic, and focal to bilateral tonic-colonic. Also for LGS, a history of tonic-clonic, atonic seizures, and more than one of the following: atypical absence, atonic, myoclonic, focal, impaired awareness, and GTC, non-convulsive SE, or epileptic spasms. There's also required that there is a development of plateauing or regression in the patient, and an EEG finding of diffuse or multifocal slowing inconsistent with an LGS diagnosis on the LGS side. All patients, the treatment-resistant countable motor seizures with an average of more than 4 observed countable motor seizures per 4-week period during the 12 weeks prior to screening while on stable ASM. The key thing here is this is an add-on therapy. So we're not asking patients to stop what they're taking. We're adding it on to what they're on, okay? And so when they're randomized, they either get randomized to what they were taking plus bexicaserin or what they were taking plus placebo. So those patients who pass screening will actually enter a titration. It's the exact same design, whether it's the Dravet study or the Deep Ocean or the Deep Sea, are the same basic design. It is a 3 times a day dosing at this stage. It's something we're working on within the company to see if we can improve that by the time we get to market. But for the study, it will be a 3 times a day dosing. Patients will titrate over a 21-day period and then be in a 12-week maintenance period. At the end of that, they'll be tapered down because we won't know who's on active or treatment, and then they'll have to retitrate up for the open-label therapy. That'll go for 52 weeks, at the end of which there will be either a continuation of the open label or some other form so patients can stay on the drug until the drug is approved. The primary outcome variable here is we're going to be looking for countable motor seizures. However, we will be looking at other things, including safety. We'll be looking at quality of life. We actually have a behavior scale within there as well. As in the phase 2, there are no echocardiograms required and we, as I mentioned earlier, the breakthrough therapy. In terms of the sites, there are 30 sites within the United States. Most of these are up and running and active at this stage. We're still waiting on some of the paperwork. And then there's a number of countries, as you can see, all the way to the right-hand side coming on board. So this is truly a global program. And if you want more details about specific countries, specific sites, my colleagues will be able to help you at the break. And if you want more information in terms of the websites and where to go about these trials, please, you can see it here, deep-studies.com. And you can read more about that there, as well as on the clinicaltrials.gov as well if you want additional information. And as I conclude, I just want to say thank you very much for your time and attention here. This partnership is really important for us. I think it's important. First of all, thank you for inviting me. And it's really important that we work together, right? That the faster we can get these trials done, the better chance we have of getting therapeutics for the patients who really need them. So thank you very much.
Thanks so much, John. And last but certainly not least, we're going to invite Kelly D. Real from Praxis Precision Medicines to talk. Thank you for being here.
Hello again. Kelly, I work at Praxis Precision Medicines. I'm the Director of Natural History and Diagnostics in Epilepsy, and I've been with Praxis for about 7 years. Our mission statement at Praxis: to help patients by delivering life-altering treatments faster and more effectively than has ever been done before, and to do it again and again. And our CEO at Praxis is constantly drilling into us that every day matters for these epilepsy patients, and how can we do things faster and quicker and still keep quality? How can we be efficient? And Praxis really is an epilepsy company. This is our platform at Praxis. We have one of the largest epilepsy pipelines in biotech. We have one program for movement disorders. All of our other programs are for epilepsy. So we have 2 programs at Praxis. One is called Cerebrum. It's our small molecule platform. And I'm going to be talking to you today about relutrigine. And then we have Solidus, which is our antisense oligonucleotide platform, or ASO platform. And relutrigine is in the sodium channel blocking class, but it is not a typical sodium channel blocker. It's a functional state modulator, which means it's selective for the hyperactive sodium channels. And regardless of what gene mutation you have in epilepsy, sodium channels play a part in seizure activity. Relutrigine is a liquid formulation, and it does not need to be titrated. And probably many of us in this room have experience with slowly titrating up over a period of weeks or even months to get to a therapeutic dose to see if this is even going to help our children. Relutrigine, the dose you start at, is the dose you stay at. It only has to be administered once per day. It has a very long half-life, as in 3 days long. So it's great for steady, even levels, and only having to administer it once per day is very nice. It can be taken by G-tube. It can be taken orally. And then it's also ketogenic diet friendly, so there's no added sugar. And the first trial in patients, so our first phase 2 trial after our phase 1 trial in healthy volunteers, that we took relutrigine into, was a very small trial with SCN2A and SCN8A patients. And we set this clinical trial up like a standard traditional clinical trial where we had study sites. And so most patients, unless they were local to a site, would have to travel to be a part of the trial. And, you know, the 2A and the 8A community were excited about this medication for, you know, a couple years before it was available for clinical trials. And I thought there would be a massive wait list as soon as it was opened for patients to be signing up. And, you know, Hillary's in this room, but at that time she was running the SCN8A Foundation. And I remember calling her and saying, where are the patients? Like, what's going on? We're ready. And she said, well, you know, it's so difficult for these families to pick up and go on an airplane and travel back and forth for these constant study visits. And she was right. And so we put our heads together at Praxis and we figured out, how do we remove travel completely for the patients and the families? How do we bring this trial to the family's home? And we were able to figure out in epilepsy how to run a completely decentralized clinical trial, so travel is no longer necessary, and every single aspect of the trial comes to the family's home. That means the physician is traveling to the family's home, a nurse is going to the family's home, medication is shipped, labs are drawn out of the home, assessments are done out of the home. And so now we have options for families. Families can participate fully remote from home, or if they want to go to a trial site, there's that option, or if they want to do a hybrid and a mix of both. And this study was designed, it's randomized 1-to-1. There was 16-week double-blind period. So on the day of screening, the team comes to the patient's home, they do all the labs, all the assessments, and then for 28 days, the caregiver is just filling out a seizure diary. And in this trial, patients had to have a minimum of 8 motor seizures in that screening period. And then they were randomized into the double-blind portion. And there was a study visit every couple weeks. And then after the double-blind portion, there was 48 weeks of an open-label extension, which just means no more placebo group. Every patient's on active relutrigine. And we did an interim data cut in this trial. And SCN2A and 8A patients, many of them have very severe frequent seizures. And so to have 8 motor seizures a month, many of them had well over that. And many of these families had been already trialing the best available standard of care. And so you could remain on your standard of care, your current anti-seizure medications. They were limited to 1 in the sodium channel blocking class. But when we did an interim data cut at the end of the double-blind portion, so those first 16 weeks, there was a 46% seizure reduction, and that was very exciting in these patient populations. And then we just recently, about 2 months ago, did another interim data cut. And at the 11th month mark, so the last patient to be dosed had reached 11 months in open label extension, there was a 90% seizure reduction, which is incredible in this patient population. For the first time ever, patients were experiencing seizure-free days and seizure-free weeks, and for some of them, even seizure-free months. There were no serious adverse events due to relutrigine, and there was no dose reduction needed of relutrigine in this trial. In addition to the daily seizure diary, seizures are the primary endpoint. We also utilized a scale, it's called the CGI, and we pulled out 4 different domains, 4 symptoms that we know is troublesome in this patient population. So we looked at disruptive behavior, and this could be anything like agitation or irritability. We looked at communication, and it's the baseline of the child. So whether the patient has no communication whatsoever, or communicates with noises or flashcards or an assisted device. We looked at seizure severity and intensity, so how long are the seizures lasting? Is there a need for rescue medication often? And then alertness, how aware is the child? Are they interacting with those around them? And this was assessed at the screening visit by both the clinician and a primary caregiver. And then it was assessed again at the end of those 16 weeks. And as you can, just after 16 weeks on drug, there was a pretty significant improvement in all of the domains. And we recently expanded to a phase 2-3 study of these SCN2A and 8A patients. And we have about 60 more patients enrolled into that study. And there's also a much broader use of relutrigine than just sodium channelopathies. So as I mentioned before, all genetically driven DEEs, the sodium channel plays a part. And so now we're taking relutrigine into a broader DEE trial, and GRIN being one of those genes where this drug could be efficacious. We've trialed relutrigine in a lot of different animal models, and I want to point a couple out. So SCN2A is listed on here, and that was the mouse model that I talked about in my earlier presentation, where a lot of different anti-seizure medications, all the best available, were trialed in that mouse model, and nothing reduced the seizures until the ASO. Praxis 562, or relutrigine, was also trialed on that mouse model and stopped the seizures, as well as SCN1A, where sodium channel blockers are typically contraindicated. It's a loss of function mutation for many patients. It was also efficacious in that mouse model, and in addition to other genetic epilepsies. And so the EMERALD clinical trial, we just activated our first site in the US last week. So we're getting ready to start screening patients for this trial. And we're able to offer the fully at-home decentralized option in this trial too. So any family that wants to participate, it's a global study, but in the US, they have the fully remote at-home option, or we have about 6 trial sites. If you're local to a trial site and you want to be seen there, or you can do a hybrid and a mix of both. But there'll be a screening visit, so if you're doing the at-home option, the team comes to your home, You have the screening day, and then you're just filling out the daily electronic seizure diary for 28 days. The minimum seizure requirement in this trial is at least 4 motor seizures per month to be eligible. And then at 16 weeks of treatment, there's a study visit once per month in the double-blind portion. There is a placebo group, and it's randomized 1-to-1. So half the patients will be on a placebo just for those 16 weeks. And then there's an open label extension that's 32 weeks long. And then any patient that's seeing efficacy on relutrigine, there's a couple years added onto the protocol, so you can go into an expanded access program. You wouldn't have to stop the drug if you're seeing benefit in your child. And then who is eligible for the EMERALD study? So if you have a documented DEE, so any gene mutation in an epilepsy-causing gene, or any syndrome diagnosis like LGS that was mentioned. It's a pediatric and adult trial, so you have to be 2 years old at the time of screening, all the way up to 65. You just have to have had your first seizure by 12 years of age. And then, as I mentioned earlier, 4 motor seizures per month. And you can stay on your standard of care. You can remain on your current anti-seizure medications to be in the trial, but no more than 2 of those anti-seizure medications can be in the sodium channel blocking class. And I've listed those out on a slide. And we make this very easy for patient families. So we have a trial-specific website. So anyone that is interested can learn more information on the website. And what you're able to do from that website is schedule a call with a nurse navigator. And she's just going to run through a few key inclusion-exclusion criteria questions, the ones I covered. And if your child is eligible and you're interested in screening, then she transfers you to a study site, and the study site will reach out directly to you, answer any questions you might have, send you the informed consent form, and then you'd be able to schedule a screening visit. And that is it. Are we doing questions? Okay, I'll stay up here.
Well, thank you guys all so much, and we're going to open up to the floor for questions. Start here. Yeah, of course.
Hi, my name is Ashley. I just have a question regarding, Kelly, the SCN2A molecule that your son is taking, and that you said that it was approved by the FDA already. And my question is regarding Radipradil, that is taking much longer to get approved by the FDA. So I just wondered, what's the difference sort of between those two?
You're talking about my earlier talk, the ASO?
Yeah.
So the FDA approved it to be in a clinical trial.
Okay.
Very different than a commercialized drug where a doctor can write a prescription.
Okay, understood. Thank you.
Mm-hmm.
Just a general question for anyone. Just curious how dosage determinations are made based on, for any of these trials.
So I can start. In our study, we look at the NMDA receptor, GLUN2B, that little subunit. We figured out in animals how much Radipradil needed to be in the blood to bind to that receptor at a level that reduces seizures. And then what we do is we figure out what dose do we have to give to children or patients that gets to that level in the blood that would lead to a binding in the receptor. And then we test those levels in humans. In the early study, the phase 1b study, we actually, every single dose level, collected blood, and we looked at the blood concentrations in every child with every dose. And then what you do is you then create a predictive model, which is like that you take that data, you look at predictions of dose ranges, and then you try to fix the dose at a level that achieves that blood concentration and that receptor binding across the whole population. And then that's the dose you carry forward. So it starts with animals. You do a link to humans, you do the early phase human study, and then you create a dose level in phase 3 that then achieves that same dose level. Is that clear?
So, but for someone in the trial, a 6-year-old in the
Yep.
trial versus a
Yeah.
15-year-old in the trial.
So what we do, so the question for those who couldn't hear, you have different ages, you have different weights. How do you manage that? We are dosing on a milligram per kilogram basis. So it's you give a quantity of drug for a given weight that would then translate to that concentration in the blood. And that's how you get that constant level across a broad range of weights and ages. I don't know, it's probably the similar. Yeah.
Hi, John. My question is with regard to Radipradil. It's a 2 to 18-year-old,
1 month to 18.
1 month to 18-year-old. Do you anticipate that this will transfer into adult medication as well, or will this be something that's specific to individuals who are that age?
So once you include an 18-year-old and 17 and 18-year-olds in the study, that is viewed by regulatory authorities, if you show an effect in that age, to then be applicable to the adult population. So the objective is to make it indicated for all ages.
Thank you.
We have one in the back.
Yeah, this is not necessarily a question, but I want to, and I hope I'm not overstepping my bounds, but this was a clarification I wanted to provide for the Lundbeck talk, was that you mentioned several times that an echo was not required, and I think that people might kind of be confused about that because fenfluramine and lorcaserin, which were both previously studied, had those off-target effects. And one of those off-target effects was that it could affect the heart valves. And so those studies needed to have echocardiograms for surveillance, and fenfluramine still needs to have cardiac surveillance for people who use it. But because this agent is more specific, you were told that you didn't need to have that same surveillance, and so that's why there's no cardiac screening built in. The other thing I wanted to provide was that, people might be like, well, serotonin, like we never talk about serotonin, that's for depression, like what's going on? And that comes, I think, also from a long-standing body of research from Dravet syndrome and zebrafish that looked at serotonin as a modulator of seizures in many of these rare epilepsy disorders. And so they then went to other kind of techniques to say, how could we use medications that are maybe already available or develop a medication that targets those serotonin receptors, because they do seem to be involved in some epilepsy some way. And I think I'd say that's because the brain only has so many neurotransmitters, right? It only has so many ways to communicate and to allow for neuronal transmission. So now we're finding out that maybe this mechanism is involved in epilepsy as well.
Thank you.
Sorry, I just wanted to clarify for everyone.
Bring it there, yeah, and then I'll get you guys over here. I'm going to give it to her because she's had an opportunity. Thank you. This one over there, then you. Okay.
Yeah, this is a question for John and Mike. Can you address family burden, family travel burden to be a part of your trials?
Yes, I can. Yes. So we have instituted a number of things to help families in terms of assisting with travel, allowing multiple family members to travel with their child to go there if they have to actually fly. There's a number of things that we've incorporated. In terms of the specific details, I would encourage you to actually go out. Our team will be at the table. Mm-hmm. And if you have specific questions about where you live and where you're going, or where you think you may have to go, they can actually give you all those details. But yes, we've instituted a lot of things to help assist with that.
Similarly, we've done that too. We have travel, we have housing, we arrange for those things. We also are implementing the EEGs that are required in the study will be done at home, so there's no overnight for that. So we've tried to do those things also, and as we roll out the study, all of those details will be clear. But the goal is always to lower the burden as best possible based on what's required in the study. There are going to be different requirements in each study, and just some of them are just have to be done in the hospital.
Hi, all. I'm a GRIN 2A mom. General question. So epilepsy's been a problem and a symptom and a syndrome for a long time. And my understanding is we still don't understand a lot about seizures and why they happen. So I wonder, like, why now are we kind of getting some of these bucket level drugs? Like, what changed? Why now? Why not 10 years ago?
Yeah. So I think we've had a lot. We've had bucket level anti-epileptics for a long time, right? I think what you're seeing now is more specificity and cleaner drugs. So one of the things with bexicaserin that I really need to touch on is the drug-drug interaction issue here is we've done a number of those types of tests, and there's no drug-drug interactions that we see here. You're not seeing that off-target piece. We're trying to get more specific. So to me, that's the change. While we're getting this bucket level, we're trying to avoid the off-target stimulation, I guess. Mike, you had any comments generally?
No, I think that's it. But I also think that with that increased specificity, there's an increased understanding of mechanism of disease. I mean, there's been an evolution in understanding not just what causes the seizures, but what the underlying defect is and how that translates into the very specific target, which has allowed this increased specificity of targeting, which, if you're targeting the underlying biology, leads to the potential to actually shift things. That's a brand new thing. When in epilepsy, where previously it was just general hit, knock down the seizures, you'll hopefully help everything else, because we don't know what's causing the disease. When I was training, I thought it was not that long ago, but I guess it was that long ago. It was all about just epilepsy's epilepsy, their seizures, and that's it. That's not the way it works anymore, so it's been an evolution.
Yeah, and I would say that the big evolution there is genetic testing, right? So that is what has revolutionized these treatments, is because, I tell families all the time, when I started testing kids, you could test for one gene, and if you didn't have SCN1A, Dravet, which was the first epilepsy gene, then we didn't know what it was, right? And so since then, we've now had hundreds and thousands of genes related to epilepsy, which has helped us to illuminate the mechanisms of epilepsy. You talk about people not knowing how a seizure starts or why it starts. Well, all of these genetic diagnoses have exposed a lot more of these mechanisms to allow us to see how they are dysregulated and then build animal models that can be used. I mean, some of these medicines are medicines that are used for general epilepsy or are now being used for general epilepsy because of the knowledge that we've gained from genetic epilepsies.
Great. Is there one over here? Okay.
Hi, GRIN 2A as well. And I'm not quite sure how to formulate this question, but our neurologist has referred her for a VNS. So the nerve stimulator. And I almost feel like we don't know what else to do. We've tried her on all of these meds, so let's just implant this device in her. So I guess I don't know how to help communicate with the neurologist about some of these things that are ongoing. It's like, we're kind of in this quandary of what's the next drug that we can try. We've tried probably 10 different drugs at this point. She's on 4. They're switching out another one. And to your point, some of them have heart. The heart is the only thing that works really well on her. So it's like we want to be very cautious and very educated on the decisions that we make, but we feel like that relationship with the neurologist is a big piece of that, but also helping them to understand that, okay, maybe before we get to that level of putting a device in her that may or may not work, like what are the other things that are out there? Again, I'm kind of rambling because I'm not even quite sure how to formulate the question for you guys, but it's a real concern of ours.
So I'll start, pass down the line. So I'm not surprised with the frustration because, again, having trained, when we were training in neurology, and I don't know the era of your neurologist, I have to be careful about my age, it becomes, everything was a name of a person and a description. That's neurology. Neurology was a description of a disease that got named after an old neurologist, and then nobody sorted it out. A lot of neurologists are still in that realm. And so the evolution of testing and the disease, the genetic testing and the evolution of a treatment, that could be something they're just not aware of. And that's an educational component that actually you are more educated in than they might be. So it's that sharing of that knowledge I think is important. Don't assume. I can tell you, having engaged with neurologists and the regulatory authorities, FDA, EMA, they don't know. It's too new. They're keeping up on every disease in the world. They don't understand this. You know this better. I always tell people, do not hesitate to share information. Do not hesitate to give them what you've learned here, because honestly, don't be afraid, because they actually might appreciate it, because they don't want to not be able to treat your child. They just may not know how. And we're seeing this commonly. Coming in our phase 1B study, and I'm sure the study you're going to hear, people are coming in 4 and 5 drugs because nobody knew what else to do. So it's not an uncommon story. And I always tell people, just tell them. Just look what I learned this week. You'd be surprised the positive reaction you might get.
And I would just add that we do a lot of work at Praxis and working with different pediatric neurologists and informing about the trial opportunities in an attempt to recruit patients into the trials. And I would say that neurologists on a whole are hesitant to refer patients into trials until something's FDA approved and known to be safe and efficacious, and they're familiar in utilizing a certain medication as a tool. What we do at Praxis is we have a lot of different tools put together that we can send to families that they can share with their neurologist so they can become educated on the molecule and the trial opportunity. And then we also have calls with these neurologists if they want to have a conversation with one of our medical doctors or PhDs and have a real science discussion and be informed about the trial.
I don't have much more to add, I think, other than thank you. I mean, you're not the only one thinking that. You're not the only one who faces that. It's a challenge, I know. And the best thing, which was said here, is be the biggest advocate you can, and don't be embarrassed, don't be afraid to bring it up, because I think, as Mike said, they need to know, they need to think differently as well. So I think that's really important.
Yeah, and I think really we all have the tools if you need it from us too. I think sort of we have that educational material, we have it available within limitations, obviously, the rules about these things, but certainly we're happy to talk to any physician who reaches out to us.
I would like to add something to that, because yes, we all want targeted therapies. We want things to help the GRIN community. It's going to take a while, but we believe we are getting there. But in the meanwhile, we are dependent upon standard care, and NVS is not a bad standard care. It can actually be very helpful, as are some of the drugs we are already using in our clients, like clobazam or clonidine, and some of these drugs are even somewhat related to the GRIN pathway, so not that strange that they do work. So the current therapies, like an NVS, don't be afraid to use it because you're waiting for a targeted therapy. Because it can actually be very helpful. And I don't know if there are people here who have an experience with an NVS in their child, but sometimes it does not work and then you have an operation, but sometimes it does add to the seizure control.
Great. I'm going to do one more question up front, and then we'll break for lunch.
Hi, me again. Dr. Panzara, I just have a question that I don't know how much you're able to speak to this, but regarding the ratiprudil and like a rebound effect and the benefits for epilepsy eventually wearing off with this drug, as so many epilepsy drugs do have this sort of rebound effect. Are you able to sort of say anything to that regarding ratiprudil?
No, I mean, what I can say is that obviously it's something we have to watch for. That's why we do the open label extension study. And I think this feeds into what I was saying about how we have our independent committee looking over time as patients are treated to make sure things aren't changing. So that is something that we're sensitive to, that we're watching for. And actually, is increasingly becoming a regulatory requirement to show one way or the other when the application is submitted. So.
Okay, thank you.
Great, thank you guys all so much. Thank you to our speakers for being here. We really appreciate all of your hard work.